Imagine you are trying to find one specific friend in a stadium filled with thousands of people, but you have a magical magnet that attracts only them. That's exactly what scientists do when they want to find one specific protein among thousands in a cell. Hey everyone, welcome back to my channel. Today we are diving into the fascinating world of immunoprecipitation, a powerful laboratory technique that uses the precision of antibodies to fish out specific proteins from a complex soup of molecules. In the next few minutes, you will see how we break open cells, let antibodies find their perfect match, and then pull those proteins out for study. Whether it's protein protein interaction, disease research, or mapping DNA binding sites, this technique is a game changer in the field of molecular biology. So, the first question arises is what is immunoprecipitation or IP? IP is a biochemical method used to isolate and concentrate a specific protein or antigen from a complex mixture such as cell lysate or serum using an antibody that specifically binds to that specific protein. This technique is based on some principle. That is, antibodies have high specificity for their antigen, which is the target antigen. When an antibody binds to its specific antigen in a solution, the resulting antigen antibody complex can be pulled out from the mixture using solid support such as protein A or G beads. Then this allows purification and further analysis for the protein and any molecules bind to it. So let's start with the steps that includes in the IP technique. The first one is the cell lysis, where cells or tissues are lysed to release the proteins, where we can find the target of interest. Here the red circle is the protein of interest. Then the second step is the antigen antibody complex. A specific antibody is added to the sample to bind the target protein. Then the third step is the formation of antigen antibody bead complex with the other proteins. Here protein A or protein G coated beads, mainly the agarose or magnetic beads, are added. These proteins have high affinity for the Fc region of antibodies, helping capture the antigen antibody complex. The next step is the wash and elute antigen antibody beads complex without other proteins. That means the beads are washed to remove the unbound proteins and the impurities. And in the elution step, the antigen is released from the antibody bead complex using an elution buffer. The final step is the purification and the downstream application. This purified protein can be analyzed by SDS page, Western blot, mass spectrometry, or enzyme assays based on what you are searching for or what analysis you want to do next. There are many types of immunoprecipitation present. There are direct immunoprecipitation where antibody is added directly to the lysate and then captured with beads. In indirect immunoprecipitation, antibody is first bound to the beads before incubation with lysate. In co-immunoprecipitation, is used to study protein-protein interaction by participating a protein along with its binding partners. In chromatin immunoprecipitation, is used to study the protein DNA interactions, and RNA immunoprecipitation used to study the protein RNA interactions. There are so many applications of this technique. So we can say that to study the protein purification, to identify the protein-protein interaction, to study the post-translational modification, investigating the signaling pathways and mapping protein nucleic acid binding sites with the help of CHIP or RIP. With every techniques, there are some advantages, like it is highly specific due to antigen antibody recognition and can insolute low abundance proteins. There are some limitations also of this technique, such as requires high quality and specific antibodies to bind, Antibody may interfere with other proteins and other things that can alter the function and interactions. There is a risk of non-specific binding if washing is inadequate. So we cannot say that every technique is perfect, but we have to do it precisely so that maximum advantages can be taken from that particular technique. So that's all for this video. I think you like this video, then hit the like button and subscribe Bioscope for such more content. Thank you for watching.